Hello, everybody, and welcome to NOAA Office of National Marine Sanctuary and Exploring by the Seat of Your Pants live interaction. Today, we are going to be exploring coral biodiversity in Flower Garden Banks National Marine Sanctuary. Before we get started and dive deeper into Flower Garden Banks, I'm going to turn it over to Joe Grabowski from Exploring by the Seat of Your Pants to give us a rundown on how today's interaction will work. Hi, Joe.
All right. Hey, Hannah. Great to be here today. Great to see everybody starting to tune in to today's webinar. So as mentioned, my name is Joe Grabowski, and welcome everyone to today's NOAA's Office of National Marine Sanctuaries event. I run an organization called Exploring by the Seat of Your Pants. So we bring science, exploration, adventure, and conservation alive into classrooms across North America and around the world. And I want to share that website with you briefly before we get started today. So if you visit exploringbytheseat.com, you'll find the website. As you scroll down a little bit, you will find a spot to sign up for our newsletter. So every week we send out a newsletter that updates with the events that are coming up. And just a little sneak peek at what we do have coming up this week. Later today, we'll be feeding piranhas at the Ripley's Aquarium of Canada. We have a trio of environment events coming up: air pollution, monitoring contaminants, as well as monarch migration. Tomorrow we'll be hanging out at the Duke Lemur Center in North Carolina, checking out some of the lemurs that live there in the forests. So we'll learn a lot there. And then if you dig a little deeper into our lessons, you'll find we have a whole week worth of ocean events coming up next week, ranging from marine biologists to virtual field trips, feeding sharks. It's going to be a lot of fun. So one more thing to share before we start today is we are using Slido to interact today. So if you've never used Slido before, we're going to have some live polls, some quizzes that you can interact with us through. So you can go to the website. It's just sli.do, and then you'll be prompted for an event code, and today's event code is flower. You can also find a direct link, which I'm about to share into the chat bar. If you just click the direct link, it'll bring you right in. And if your cell phone is handy and you've got a QR scanner, I've got a code up here that you can scan that'll bring you right into the room via your cell phone. So we'll be doing some live polls today you can interact in. We'll also have a quiz during Josh's presentation as well, and you can take part in that. So a few ways you can join us for the live
Joe,
slide.
I am unable to hear you.
Really?
That might just be me.
Okay.
Josh or Kelly, are you able to hear Joe?
Yep, I hear you just fine, Joe.
I can hear and see Joe.
Okay. All right. We'll assume that it's going through okay. I'm going to come back from that share. Let me stop that there. There we go. And so today we're going to take a virtual field trip to the northwestern Gulf of Mexico with expedition lead Dr. Joshua Voss from Florida's Atlantic University's Harbor Branch Oceanographic Institute. Before we do that, though, I'm going to throw things back to Hannah McDonald, education specialist with NOAA's Office of National Marine Sanctuaries. She's going to take us on a little virtual tour of the marine sanctuaries around the country. Hannah, how about now? Can you hear me?
Yep, I had to take out my headphones. I apologize about that. All right, so yes, like Joe said, I'm going to take us on a virtual tour of NOAA's Office of National Marine Sanctuaries. The National Marine Sanctuary System is a network of 14 national marine sanctuaries and 2 marine national monuments. We encompass over 600,000 square miles of marine waters in the United States. So as you can see, we span from all the way in the Northwest to the Pacific and many sites in the Atlantic as well. In a minute, I'm going to go take us on a virtual photo tour of these sites. But first, I want to engage you all with a few Slido questions. So if you missed the information that Joe shared at the very beginning about how to get to Slido, no worries. The link to Slido is right in the chat on the control panel. So you can click in there and then enter your answer into how many people you are watching this live stream with.
Okay, so just a quick reminder, you can also find it at sli.do and then use the event code flower. We have a few starting to join in, over a dozen now. It's also in the chat link, but let's take a look at the results so far. We've got a lot of solo viewers and one group of 5 so far today.
Awesome. Well, welcome everybody. Our next Slido question is, have you ever heard of NOAA's National Marine Sanctuaries before?
All right, this is our best number yet, Hannah. 100% of
Wow.
the group so far has heard of a sanctuary before.
That is awesome. I'm glad to hear it. Hopefully these are returning viewers too. So as I said, we're going to take a virtual tour through our National Marine Sanctuary System, starting first in the most northwest corner of continental United States in Olympic Coast National Marine Sanctuary. As you can see in this photo, they have very lively tide pools. Going further south, we have Greater Farallones National Marine Sanctuary off the northern coast of California. Connected to Greater Farallones, we have Cordell Bank National Marine Sanctuary, which protects some deep sea corals. A little bit south of that is Monterey Bay National Marine Sanctuary, where other expeditions have found some incredible ocean research findings, like this brooding octopus garden. And our last sanctuary in California is Channel Islands National Marine Sanctuary, which is about 25 miles off the shore of Santa Barbara, protecting lush kelp forests. Going further west into the Pacific, we have Papahanaumokuakea Marine National Monument. Now, this is the largest marine conservation area in the world. We also have Hawaiian Islands Humpback Whale National Marine Sanctuary, which protects the breeding grounds for humpback whales. And then even further the Pacific, we have the National Marine Sanctuary of American Samoa. And here we protect one of the longest living and largest known coral heads called Big Mama. And Flower Garden Banks, coming back to continental US, 100 miles off the coast of Texas. Here is where we are going to explore today and learn all about coral biodiversity. Off the coast of Florida, we protect the Florida Keys Reef Tract and Florida Keys National Marine Sanctuary. Off of Georgia, we protect a live bottom reef ecosystem in Gray's Reef National Marine Sanctuary. Our very first National Marine Sanctuary ever designated is the Monitor National Marine Sanctuary off the coast of North Carolina. Now, this sanctuary protects the USS Monitor, a Civil War era shipwreck. And to our newest National Marine Sanctuary, we have Mallows Bay Potomac River National Marine Sanctuary in the Potomac River in between Maryland and Virginia. Now, as you can see from this photo, it's quite a unique ecosystem. Here we have partially submerged shipwrecks from the Ghost Fleet. Going further north in Massachusetts Bay, we have Stellwagen Bank National Marine Sanctuary, known for incredible whale watching. And even in the Great Lakes, we have Thunder Bay National Marine Sanctuary, protecting over 200 shipwrecks. And here we have our next Slido question. Since we just went on a virtual tour of this system, I want to know if you've ever visited a National Marine Sanctuary before.
All right, so lots more have found the Slido room. Let me check our results on that question. 66% have visited a National Marine Sanctuary.
That's great. That's a very good number. Many people knowing and visiting sanctuaries today. And now that you have a better idea of the places, we are going to go over a little bit of what sanctuaries do. So we protect things like the sea giants in Monterey Bay National Marine Sanctuary, to the small sea life like corals and reef fish. We protect places with abundant biodiversity and places that provide shelter for some of the most charismatic marine species, like this endangered Hawaiian monk seal and this green sea turtle. And as I mentioned in some of the sites, we also protect maritime heritage like shipwrecks in Thunder Bay National Marine Sanctuary. We are mandated through the National Marine Sanctuaries Act to protect these resources for generations to come. We do that through restoration projects as well as education and outreach, like this program you're participating in today. So these are your special marine places. These are your places to enjoy and explore. These are your places to paddle, to fish, to snorkel, to boat, and to surf. All types of recreation are welcome in the National Marine Sanctuaries. Ooh, so here we have a quiz question. And I want to know, I've only mentioned it once, if you can recall how many square miles the National Marine Sanctuaries encompass.
All right, just before we answer that question, I want to remind everybody that during Josh's presentation today, we will have some quiz questions. So right after we finish this poll, I'm going to shut down the survey and open the quiz. So you just have to pop your name into your quiz spot and you'll be ready to go. And then we'll talk about those when the first quiz question comes. But 79% of people went with 600,000 square miles.
Nice. Everyone is right. And so now, before I turn it over to Dr. Joshua Davis from Florida Atlantic University, I want to talk about what brought him to this project. So this was awarded, this is money awarded from NOAA, the National Oceanic and Atmospheric Administration, to organizations who are willing to do telepresence exploration. Now, what is telepresence? Telepresence is being able to bring the ship to shore. Now, you can do that to scientists or to education venues. So part of Josh's work this summer was to bring and broadcast his work with coral biodiversity live to audiences around the country. So that was awarded to the Global Foundation for Ocean Exploration, who Josh worked very closely with Flower Garden Bank's National Marine Sanctuary staff. And with that, I'm going to turn it over to the lead scientist of the Coral Biodiversity Leg, Dr. Joshua Voss. And I am going to, I'm not able to click on the control panel. Joe, would you be able to make Josh the presenter?
I can do that. Let me jump over to the dashboard.
Thank you. Something has locked me out on my computer.
All kinds of adventures today. All right, Josh, that should be good for you.
All right, thank you very much, Joe and Hannah. Can you guys see my screen and hear me okay?
Yeah, looks good.
All right. Switch over to slideshow mode and get rolling here. Thank you all very much for taking the time to stop by today and learn a little bit about one of my absolute favorite places in the world, Flower Garden Bank's National Marine Sanctuary. I'm a professor at Harbor Branch Oceanographic Institute, which is a research institute of Florida Atlantic University. We're located in Southeast Florida along the coast, but I've worked in the Gulf of Mexico with Flower Garden Banks for a little more than a decade now. It is one of the most incredible places in our sanctuary system, and I'd argue probably one of our most impressive marine habitats in U.S. waters, period. In addition to my role as a faculty member here at FAU, I'm also the executive director of a NOAA cooperative institute, the Cooperative Institute for Ocean Exploration, Research, and Technology. This is a project that is a collaboration with NOAA's OER office, led by Harbor Branch FAU in collaboration with UNCW and University of Miami. And we have four primary tasks. The first is to explore continental shelf frontiers, so areas out on the continental shelf that have high potential interest and interesting ecology. One of the areas that's our primary focus are vulnerable coral and sponge ecosystems. We know that coral and sponge ecosystems are incredibly important, but we also know that many coral ecosystems are under threat. And so any strategies that we can employ to understand and protect them are very important. We're also tasked with developing advanced underwater technologies to increase the scope and pace of underwater exploration. And then finally, we play a big role in training the next generation of ocean explorers, hopefully like some of you on today's presentation. So we use a whole bunch of different tools and techniques in order to access the ocean habitats and explore them. Everything from sonar multi-beam and surface gliders to underwater autonomous vehicles, submersibles, and ROVs. I still would argue that getting people in the water, particularly divers who can see things with their own eyes, is an important component of exploration as well. And so most of the activities that we do are supplemented by divers in the water at some point when those are in habitats that we can physically access, given the limitations of our bodies. Part of our process is not only the exploration and the data collection, but also then building the community and telling those stories afterwards. So part of our training for graduate students through our Cooperative Institute is to not only get them out at sea and get them involved, but then to have them tell their stories and share that information in multiple settings, be that normal scientific conferences or small school groups as well. And with regard to coral reefs, we're really trying to build understanding in a couple of key areas. And the first of those is trying to find and characterize new coral reef resources. So in particular, we know that as most of the coral reef resources that we are familiar with are shallow coral reefs, areas that are within diving depths down to about 120 to 130 feet. And that's primarily because they've just been easier to access over time. However, as we start to explore more deeper areas of our coastlines, we're seeing that there's more and more coral reef resources that are out there. And it's an open question about how do we best protect them and what resources might they provide. One of the interesting things about discovering and investigating these new places is identifying new species or perhaps new records of species for certain locations. And some of the areas that we're finding the most novel species are in sponges and other small invertebrates. So I'll give a couple examples of that today. And then what I'm really interested in fundamentally in terms of research is how do biodiversity and connectivity among habitats drive resilience and persistence of marine ecosystems? In other words, how can we understand what has made these immense and beautiful ecosystems persist over time? And what can we do to ensure that they persist into the future? And so part of that is taking the data, information, and images that we generate, and then providing knowledge and even predictions about what might happen in the future to help improve coral reef management, both nationally and internationally. So for those coral reefs, most of you in your mind's eye, when you think about what a coral reef is, are probably thinking about a shallow coral reef in that range of 0 down to about 40 meters or 120 feet deep, where there's lots of biodiversity, there's lots of growth. In many cases, the coral cover can exceed 50 or even 80% in some areas of the world. Lots of fishes, lots of rugosity and habitat that make these incredibly productive and biodiverse habitats. But once you move beyond that recreational depth limit, once you start to get below that 30 to 40 meter or 100-foot horizon, we get into these mesophotic reef areas. And these are incredibly interesting reefs where we start to see corals plate out in huge amounts of area. And these mesophotic reefs are an area that have become increasingly interested, or sorry, have become an increasing interest for scientists recently, simply because we're developing better tools to access them more frequently. Advances in technical diving, advances and reduced cost in terms of ROV use have allowed much more access to mesophotic habitats than we've ever had before. So we know where mesophotic habitats start, right around 30 to 40 meters, but they extend down to around 150 to 200 meters, depending on where you are. And so my first Slido quiz question for you today is here. Why do mesophotic reefs stop around 150 to 200 meters?
All right, so most have signed up already to start the quiz, so I am going to start it now. So jump into Slido. Each question you have 20 seconds on the clock. The quicker you answer, the higher your score. And then, of course, being right is pretty helpful as well. So here we go. First question is up. What environmental factor is the primary driver of most coral reef depth distributions? So we have pressure, light, temperature, or nutrients. We've got 10 more seconds. All right, 64% went with light.
Fantastic. That means 64% of you are correct. Well done indeed. So the major limitation at the bottom of mesophotic is the limit of the photic zone. So mesophotic just means middle light. So these are areas that have slightly less light than we might see in a normal shallow reef, but still enough light to support photosynthesis. So remember, for many corals, they have these symbiotic algae that are living inside their tissues, allowing them to do photosynthesis and functionally act kind of like a plant. They can supplement their photosynthesis by doing feeding as well, but they can be much more productive because of that relationship with their symbiotic algae. Below 200 meters, we get into deep reef areas, and they still calcify. They can still make hard reefs, but below that limit of the photic zone, you really don't see corals that have that symbiotic algae, or zooxanthellae as they're sometimes called. So that's a pretty interesting biological breakpoint. And the combination of corals with the symbiotic algae really drives increased productivity, increased diversity, and increased abundance overall in those mesophotic depths. Now, these mesophotic reefs, as we've started to look for them more, we're finding more and more that they are extensive across the Gulf of Mexico. So through our Cooperative Institute and partnerships with NOAA Sanctuaries and NOAA OER, we've been able to conduct about 19 different research cruises across the Gulf of Mexico in the past 8 to 9 years. And essentially, most of the places we've looked expecting to find mesophotic habitat, we've found it. So mesophotic habitat around flower garden banks was already known, and there was a bit known about what was off Southwest Florida. We've done extensive mapping in both of those areas to expand those known areas of mesophotic reefs and characterize them further. We recently completed a circumnavigation of the entire Cuban archipelago and found that mesophotic reefs were extensive all the way around Cuba and may be an important contributor to overall Gulf ecosystem dynamics. And what's really interesting is that it's not just flower garden banks themselves that have these mesophotic reefs. It's areas that extend across the continental shelf margin in the Northwest Gulf of Mexico. And that's what I'm going to focus on today. Part of the reason that Northwest Gulf of Mexico biodiversity is so immense is that it's a relatively stable, warm body of water with amazing habitat on the bottom. And so if you compare the biodiversity at flower garden banks to other places, it's relatively quite high for things like invertebrates, algae, and fish, but it's somewhat low in terms of coral species. So there's 24 hard or stony coral species that are known at flower garden banks, as compared to somewhere north of 65 to 70 for the Caribbean basin overall. But even though there's relatively low coral species diversity, the relative abundance of corals and the cover of corals at flower garden banks is incredibly high. So flower garden banks has on the coral caps upwards of 50 to 60% coral cover. And for comparison, Florida Keys National Marine Sanctuary only has about 3% coral cover. There are other places in the Caribbean that also have really high coral cover, locations in Cuba, locations in Bonaire and Curacao, but flower garden banks is really one of these just spectacular habitats when it comes to coral. Now, the current flower garden banks National Marine Sanctuary just encompasses 3 banks. So the tiny one over here on our left, number 1, that's Stetson Bank, but the main 2 banks, 2 and 3, are east and west flower garden banks respectively. And these are areas that have been protected and remain protected. However, there's also been a recognition that additional banks along this continental shelf margin harbor incredibly important habitat that may be worthwhile protecting as well. And so currently, Flower Garden Banks National Marine Sanctuary is undergoing an evaluation of an expansion process. And the Sanctuary Advisory Committee has identified a number of different potential alternatives of how Flower Garden Banks might expand and what it might protect. And the preferred alternative, or alternative 3, would take Flower Garden Banks from just over 50 square miles to almost 400 square miles in terms of total area protected. So an increase of somewhere between about 6.8 to 7 times the original area of Flower Garden Banks National Marine Sanctuary. And so these areas, many of them have been explored before. That's how they've been identified as high value habitat worth protecting. But we need additional information at these banks for expansion to continue to build the case that these are important habitats that are worth sanctuary protection and conservation. And so that's part of where this project developed from. Using an opportunity to get out to some of these banks that are proposed for expansion and share their wonder with people via telepresence. So there were 3 5-day expeditions that were run back to back aboard the Sanctuary's vessel, the RV Manta, that you can see here on the bottom of the screen. My group was leading leg 1. So we're all from FAU Harbor Branch. The Sanctuary Superintendent, GP Schmal, was out there with us. We had a fantastic team of engineers and pilots from Global Foundation for Ocean Exploration. And they had also brought along this new flyaway telepresence system. So this was a really neat combination of the ROV Yogi, which was a relatively small ROV, or remotely operated vehicle, that was developed for use in Yellowstone Lake, but adapted for saltwater exploration. And then that combined with a small portable satellite communication system was really the first test of creating a flyaway ROV-based telepresence system. And what I mean by flyaway is the entire system came in a container, was delivered to the ship, moved onto the ship and set up in about a day. And then we were able to beam live from the ship to anyone that was on the internet, live images of what we were seeing on the bottom using the ROV, as well as live images from the deck of the ship as well. And Dave Lavalvo is the lead at GFOE, and he was the PI on this project overall. Hopefully some of you got a chance to catch the Leg 2 presentation that was given yesterday by Dr. Sarah Davies. And there'll be one more on June 5th by my friend and colleague, Dr. Mercer Brugler, who'll be talking more about the deep reef and black coral areas that were explored on this expedition. So for leg 1, we had some particular objectives. We had done previous transects in a lot of these areas, but we wanted to supplement those transects to further characterize those benthic or bottom coral communities, as well as assess potential changes over time. We wanted to identify, sorry for the typo there, that should say new species records. So either new species, novel species that are unknown, or species that had not been previously recorded in the Northwest Gulf. We wanted to collect samples of target coral and sponge species to assess genetic connectivity. So since all of these organisms are attached to the bottom, the way they move from location to location is by dispersing larvae, and by looking at the relatedness of corals that are on the bottom, you can get an idea of where those larvae may have come from and what the source and sink populations across the region are. And then lastly, we wanted to leverage that telepresence capability that we were bringing out with us to directly connect with classrooms as well as shore-based scientists. So here you can see on the top left, this is ROV Yogi on the back deck of the Manta on a beautiful evening. It's a system that using some input from us, we adapted the sampling characteristics to really suit coral collection and sponge collection quite well. And GFOE was very adept at helping to develop a system that could meet the needs that we needed. Here on the bottom left, you can see what it looks like inside the control room during a dive. So panels of multiple different screens that allow the operators and the scientists to see what's going on in real time, and then to make decisions about which way to go, what to investigate, and what to sample. Here you can see GP. So GP is really great fish expert and was able to help identify a lot of fish out there, as well as just general biodiversity, and give context to what the goals for the sanctuary were overall. Some additional folks from my team were Aaron Schilling in the background, Ryan Eckert, both of whom are graduate students of mine, and Jim Masterson, who's a fellow faculty member at Harbor Branch as well. And during this expedition, we ran multiple events to shore-based teams, including interacting with about 90 students that are here at Harbor Branch, both high school students and college students. And then we also had a team of about 10 shore-based scientists and graduate students that were watching live in real time and contributing to the mission by helping us to identify organisms, choose samples. And that's really, those are two of the biggest benefits of these telepresence systems, is to draw in a bigger audience and to draw in more expertise to accomplish the scientific mission. So the banks that we hit during leg one were Saunier Bank, Alderdice Bank, McGrail Bank, and Geyer Bank. And I want to give you just a kind of a feel for how these looked. So I'm going to toggle over to a video here. And if you want to see any of the videos from this expedition, you can go to the GFOE videos page, and you can see highlight videos from each of these expeditions, as well as some other expeditions that GFOE has run recently. So these mesophotic reef areas can be quite variable. So you'll notice that some of these are covered in crustose coral and algae, this kind of pink background. Some have macroalgae, but then in most cases, there are some very large corals and very large sponges. Here you can see it's collecting a small sample of a sponge species. Here we're collecting a starfish species, and this was one of the larger star corals that we observed spawning during this mission. One of the reasons that we're really interested in these mesophotic habitats is because they seem to support really high fisheries abundance, particularly important snapper and grouper species. And these sponge systems are amazing as well. Another example of one of the new species records that we observed for the Northwest Gulf was this small little long snout seahorse here. And this was one that has never been observed in the Northwest Gulf before. All right, so Joe, why don't we stop there and we can swap over to Slido 2 and take another quiz question.
All right, sounds good. So those who took part in the first quiz question, we're going to open up the second question. So jump back into your Slido area. I'm going to give you a couple seconds to do that, and then I'm going to bump it over. Again, remember, speed helps your score. So here we go, sliding over to the next question. If the preferred expansion plan for Flower Garden Banks National Marine Sanctuary is adopted, approximately how much will the sanctuary grow? Is it 2 times, 3 times, 5 times, or 7 times? Got about 10 more seconds. Numbers bouncing around like crazy. Let's see where we end up here. All right, so quite a mixture, Josh, but 38% settled on 5 times. What do you think about that?
They're close. So it's actually going to be about 6.8 times the size of the existing sanctuary, going from just over 50 square miles to just over 380 square miles. So that process is still ongoing. There's going to be additional opportunities for the public to weigh in. So if If you are compelled by this and other presentations to help protect Flower Garden Banks National Marine Sanctuary, I urge you to use your voice, share this information, and ask others to use their voices to help conserve this amazing habitat as well. All right. So let's talk a bit about what we've seen during these expeditions and previous expeditions as well. So tons of work by the sanctuary staff and many other researchers, including ourselves, have been investigating the coral reef caps at Flower Garden Banks for decades. And again, these areas have extremely high hard coral or stony coral cover, upwards of 50 to 60% on average. They have very low macroalgal cover, so that indicates that there's not a lot of competition for macroalgae. There's probably not a lot of nutrient pollution. And hard corals really dominate this system, both in terms of the biodiversity and the topography. In contrast, mesophotic reefs have about 3 to 36% cnidarian cover. And that's really a mix of hard corals, soft corals, and black corals. So you still have some areas that have relatively high hard coral cover, like this image you see in the upper panel on the right. But then you also see an increase in these anipetherians or black corals that don't contribute skeleton to the bedrock, but they do serve as important fisheries habitat and are important contributors to biodiversity overall. One of the other interesting things is that we seem to see a slight increase in sponge abundance as you start to move into these mesophotic reef areas as well. And there's recent speculation that this may be a pattern that's observed throughout mesophotic reefs, not just in the Northwest Gulf of Mexico. We've got a number of new species records. So I already mentioned the long-snout seahorse. And not only was this a new species record for the Northwest Gulf, I think this was the first seahorse that has been observed in the Northwest Gulf, period. So that was really, really cool and interesting, and a lesson in slowing down and taking a close look at these interesting habitats can allow you to see even more of the intricate biodiversity that could be present in them. And also, as I mentioned before, we think there's a lot of work to be done in terms of sponge diversity overall. So there's two individuals that we observed and sampled during this mission that we believe are probably new species. There's a dendrilla, this kind of very spiky looking sponge here in the middle that does not seem to be identifiable to any known species. And then there's also this other Zestispongia that we're affectionately calling Zestispongia bebe, or little Zestispongia, that we believe is an undescribed species. We still need to do more work to see if it's truly distinguished from Zestispongia muta. And this is a sponge or a morphotype that we've also observed on mesophotic reefs in Cuba. So it may be a species that could be unique to mesophotic habitats potentially. One of the other really interesting things that we observed is that mesophotic coral reefs just generally appear much healthier on average than shallow water reefs. So most shallow water reefs at various times are exposed to bleaching or loss of those symbiotic algae or diseases of various kinds. In all of our coral surveys across the Northwest Gulf of Mexico, including last summer's expedition, we've seen almost no coral disease in these mesophotic areas, less than 0.02% of all colonies that we have observed and surveyed. So this is much, much lower than disease prevalence values that we might see on shallow reefs, which typically are kind of a background of 1 to 3%, and can go up from there during outbreak scenarios. So this suggests that there's something about these mesophotic corals that may be allowing them to be healthier overall. And so since we know many shallow reefs are being degraded, one hypothesis is that these mesophotic reefs might serve as refugia and help to reseed shallow degraded reefs. And then it's kind of an open question about, are these mesophotic reefs more resilient? So in the face of change, be that an episodic event like an oil spill or a hurricane, or slow change over time like climate change, are these mesophotic corals going to be more or less resilient than their shallow water counterparts? Part of this question of do mesophotic coral reefs serve as refuges and can they reseed reefs can be investigated by looking at coral connectivity, by trying to see evidence of previous instances of mesophotic reefs providing coral larvae to shallow areas or providing coral larvae to one another to help improve resilience of the systems overall. So we've used one coral species to date, Montasteria cavernosa, or the great star coral, to try to investigate this question across a really broad range in flower gardens, Southwest Florida, Southeast Florida, and Cuba and Belize. And take-home story from these is that the vast majority are relatively well connected, with the exception of two areas. Pulley Ridge seems to be an outlier with a very closed remnant population, and the shallow Montasteria cavernosa or star coral populations in Belize seem to be relatively disconnected as well. So flower garden banks is incredibly well mixed. The mesophotic corals and the shallow corals are related to one another, indicating they're likely functioning as one big group and sharing larvae back and forth at different times. There's good evidence for moderate connectivity between flower garden banks and mesophotic corals in Belize, but not shallow corals, and good evidence for connectivity between flower garden banks and over into the dry tortugas in Florida Keys, but again, not Pulley Ridge. And then some more recent work we've done also shows that mesophotic corals in Belize are directly related to shallow corals in Southeast Florida and corals in Cuba as well. So there's good evidence that overall these systems are helping to support one another, and that over generations, larval movement from place to place is what allows these populations to persist and provide the ecosystem services that they do. But that also means that in terms of conservation planning, we probably need to be thinking about not just isolated sanctuaries, but networks of sanctuaries that perhaps have to involve multiple different countries and jurisdictions in order to manage them effectively. All right, so I'll pause there, and Joe, let you get to Slido question 3.
All right, this is our final quiz question. I'll give everyone a second to switch back over, and then we'll check the results and see who was sharpened on the ball. I should make a reminder as well. We'll start the Q&A soon. You can use the question box on the side in your GoToWebinar control panel, or you can send in questions via the Slido room, and you can upvote other people's to the top if they are similar to your questions. So let's get that final quiz question going. Mesophotic coral reefs are important because they have relatively high biodiversity, support important fishery species, may help reseed degraded shallow coral reefs, or all of the above. Just under 10 seconds left. All right, good job, everyone. 88% rolled with all of the above. And let me do a quick little tally here of our leaderboard. Who we have? All right, we've got a three-way tie for first place between Susan, John, and Rachel. 3 out of 3 in 22 seconds. Coming in second is Greg, 3 out of 3 in 27 seconds. And then we've got Liana, 3 out of 3 in 28 seconds. Good job, everyone. And thanks for playing in the Slido room today.
Well done, everyone. Very impressive indeed. So I'll leave you with a few kind of closing thoughts and let you know a little bit about what we're going to be doing next. First, coral reefs in general, and mesophotic coral reefs in particular, are really ecological and economic powerhouses across the entire Gulf of Mexico and are important habitats that we need to protect and conserve overall. The more and more we look for these mesophotic coral reef habitats, we're finding them. They're extensive, they're relatively healthy, they appear relatively well connected across large areas of Florida, the Gulf of Mexico, and even down into Mesoamerica. And there's efforts to protect these areas through expansion plans in flower garden banks and a revised management plan in the Florida Keys that is under consideration right now as well. So my advice to you would be to learn about those plans and to support them. These are your sanctuaries. If we want to see these resources persist for generations to come, they need your support now. Our next steps are going to be to start expanding a lot of the connectivity studies that we're doing in the Flower Garden Banks region. And that's going to involve going from 1 coral species to 10. So we'll be adding one of the species that Sarah Davies talked about yesterday, the lesser star coral or mountainous star coral, Orbicella. We're going to be adding a couple sponge species, a couple of fish species, and some black coral species as well. All of that's going to be part of a project that's funded through NCOS and in collaboration with Santiago Herrera at Lehigh and the Flower Garden Bank staff as well. And then we also, through our cooperation with NOAA OER, are going to be conducting another cruise this coming summer, next summer, thanks to COVID, where we're going to be trying to go back and look at some of these interesting sponges to look for novel pharmaceuticals. So because sponges are trapped on the bottom, they do a lot of chemical warfare to hold their place on the bottom. And many of them have interesting anti-tumor, anti-malarial, and other anti-cancer properties. So with that, I'll encourage you to follow up and learn more about the work that we're doing and Flower Garden Banks and GFOE in general. So here are a few links that can help you to find information. If you're interested, you can follow our work and Harbor Branch's work on Twitter. And I'll say again, thank you to Hannah and Joe, and then all of our friends at Flower Garden Banks National Marine Sanctuary and GFOE who helped make this expedition possible. I'll be happy to answer any questions you guys might have.
All right. Great, Josh. Thanks so much for an awesome presentation. And it looks like an amazing place to explore. I can see why it would hook and draw you in to keep doing that exploration. And that, that pharmaceutical search using the sponges is really exciting. I think there's a lot of really cool things that can come out of that. And sponges are just cool in general, being animals with like no eyes and hearts and brains. Pretty darn cool. All right. We've got lots of questions coming in the Slido room, so you can use the Slido room. I'll also keep an eye on the question bar. And let's start off, well, the most upvoted question so far. This is from Jack. And Jack's curious about the impact that sunscreen can have on coral reefs while snorkeling. And then I guess just to add to that as well, I guess the Mesophotic Reefs might be a little safer from that threat.
So I'll kind of answer the second part of that first. So part of the reason we think Mesophotic Reefs are healthier is because they are simply just more removed from any anthropogenic threats. They're less exposed to temperature extremes that might be related to climate change. They're less exposed to nutrient runoff in general because they're farther from shorelines. They're typically less exposed to fishing, but that's not always the case. Fisheries impacts can be an issue and need to be managed. I would say sunscreens on Mesophotic Reefs is probably one of the absolute least concerns that we have to worry about. There's no one snorkeling down to 150 feet. In general, the research that has been done regarding sunscreens and impacts on corals is quite minimal. There's really only been 3 published studies that have investigated this. There has been some evidence that oxybenzone, which is a component of sunscreen, can cause impacts to coral larvae. But in general, I think it's an area that more research needs to be done. As a practitioner who's in the water all the time, I want to choose a sunscreen that's safe for me and the corals. And so, alternatives are out there, and it's kind of a precautionary thing. While we may not have extremely strong evidence that sunscreens are detrimental, if we can make a easier choice that we know is not causing any problems, why not?
Okay. Another question that's come up here is, you talked about expansion of the sanctuary. Would that expansion have an impact on commercial or recreational fishing in the area?
It would have an impact on commercial fishing in the area. It would also potentially have an impact on future plans for oil and gas exploration in the region. And Kelly, I'll invite you to correct me if I'm wrong, but I don't think it'll have much impact on recreational fisheries. To my knowledge, there are no plans to close those expanded sanctuary areas to hook and line recreational fishing. Is that correct?
That's correct. We are currently in a public comment period and have a lot of information available for that on our website, and I'll put the web information in the chat box.
Awesome. Thank you, Kelly.
All right. Let's see. Albert is wondering about coral being multicolored. Why? Can you give us any insight into the different coloration of coral? Why are there so many different colors?
Sure. So corals get most of their pigments, but not all, from their symbiotic algae, especially stony corals do. And the variations that you can see in the pigments of the corals are usually related to the different photosynthetic pigments that those algae may have. Excuse me. So the predominant one would be green in chlorophyll, but there's lots of other phycoerythrins and cyanins that can cause different shades of brown, green, yellow as well. And then in soft corals, you see a lot of different changes in coloration that are related to proteins that contribute to the bright colors you see on some of the soft corals.
Okay. So a couple questions, actually it's coming up both in the question section as well as the Slido. So this one, let's see, is from Eric as well. So Eric's wondering about the events around Deepwater Horizon several years back. Did that have a large impact on those reefs?
So Deepwater Horizon had some significant impacts on deepwater reefs in the northeastern Gulf of Mexico, particularly areas that are quite a bit deeper than these mesophotic regions that I was talking about. There's been documented impacts on soft corals in that region, down to thousands of meters in the vicinity of Deepwater Horizon. We conducted a cruise shortly after Deepwater Horizon looking for impacts of oil along the southwest Florida shelf, so down the coast, the west coast of Florida, along the margin that goes towards Pulley Ridge. And thankfully, we found very minimal impacts, or really almost no detectable impacts at that time in that region. And the oceanographic conditions during the time of Deepwater Horizon, thankfully, were favorable so that there was not a large transport of oil and dispersant westward to Flower Garden Banks. So there was not major impacts at Flower Garden Banks that resulted from Deepwater Horizon. But Deepwater Horizon is a really important lesson that oil and gas exploration and extraction do carry risks. And so those risks need to be managed, and part of management of that risk is understanding what resources may be affected and developing strategies to prevent those resources from being lost in the future.
Okay. So this question's come up a couple times. I'll grab this from Dennis. Curious about scuba diving, recreational opportunities in the sanctuary. Is there opportunities for that kind of activity?
Absolutely. So those coral cap areas start only at around 55, 60 feet. And so those are totally accessible to kind of slightly experienced scuba divers. The advice for a novice scuba diver is to get some dives under your belt in the 30 feet or shallower range first. But then after you've got some experience, you can go on down to some of those 60 to 100 foot regions. And they are extremely accessible at Flower Garden Banks once you get out there. So it is 100 miles offshore, but they're accessible by private boat. There's some charters that run out there, diving charters that run out there as well. And then there are mooring balls that have been installed at East and West Flower Garden Banks that allow boats that are visiting to, number one, access kind of the areas that the sanctuary knows are the best places for diving, as well as prevent any impacts from anchoring by the use of mooring balls. So if you have the opportunity to get out there and you have the qualifications diving certification wise, I would very much encourage you to do so. It is an absolutely incredible resource.
Okay, and there was a question from Stephen. It was exactly that question about mooring being used to limit damage by anchors, and so that's taken care of. Good stuff. Another question here about the coral species between the shallow and the mesophotic. So wondering if there's much overlap in species, and could seeding go both ways? Could the deep seed shallow or shallow seed the deep?
The relative overlap in mesophotic versus shallow species is variable in different regions of the tropical Western Atlantic. It's fairly high in the Flower Garden Banks region. So of those 24 species that are found up on the shallow coral cap, we've documented at least 17 of those species well down into the mesophotic depths. But the relative abundance changes quite a bit. So things like greater star coral or blushing star coral are highly abundant in mesophotic, whereas other species like some of the brain corals are functionally almost absent once you get down into the mesophotic. In terms of could shallow corals provide larvae downward? Absolutely. There's the possibility of those larvae spreading and going both ways. Most coral larvae are somewhat positively buoyant after they are released, and then they ride on currents for a period of time until they're competent, and then start to search for and settle down in a given spot. The reason we talk about that mesophotic to shallow connectivity from a management perspective is that we know that shallow reefs in a number of areas, particularly in South Florida, have been degraded and impacted quite a bit over the past 30 to 40 years, and especially with the outbreak of a new coral disease in the past 5 years in South Florida. So anything that we could do to help those shallow areas to recover is worth our effort and interest. And that's why understanding if mesophotic corals can provide those larvae to the shallow reefs is a really important thing to consider.
Okay, so we have lots of students tuning in today, and this question's come up quite a bit. Can you tell us a little bit about some of the other marine life that makes homes around the reefs? Sure.
So Flower Garden Banks in particular has amazing fisheries species and some pretty awesome marine megafauna. As part of a mapping project we did, we were looking at the relative abundance of things like snapper and grouper, things that we like to eat in general. By we, I just mean humans. There's some really cool marble grouper populations, black grouper populations. A number of grouper and snapper populations are known to have spawning aggregations where they come together to make babies on Flower Garden Banks and surrounding banks. And then beyond that, there's extensive use of the sanctuary region by manta rays, whale sharks, tiger sharks, basking sharks, a lot of these large, big, transient planktivores and predators that kind of operate on the scales of hundreds of miles as they move back and forth among these banks.
All right, let's see if we can squeeze one more question in here. Go back to the Slido room. There we go. Okay, so this question here is just kind of a general question about, okay, I'm going to switch actually to this one. So can you give kind of a quick summary of steps that need to be taken to increase the size of the marine sanctuary? So I think they're just curious about kind of the chain that follows to get that done.
Got it. So it's something that kind of starts with building the scientific information. So developing maps and criteria for which of the areas have important marine resources. Coral reefs in particular are important in terms of supporting essential fish habitat and supporting those fisheries. So that's one of the major criteria. Whether or not other endangered, threatened, or important species may be there is part of the criteria for whether or not a region should be protected. And then in terms of the process, so every marine sanctuary has a Sanctuary Advisory Council. And in this case, the Sanctuary Advisory Council has been reviewing and discussing the proposed alternatives. And at this point, it has gone out for public comment multiple times at different levels. And there'll be additional opportunities for public comment. Ultimately, for additional protections to be put in place, it would need to be approved both by that council, by sanctuaries overall, by the Department of Commerce, and by the president.
Okay. Well, first of all, I want to give a shout out to everybody who tuned in to today's event. Thank you so much for joining us. Thanks for sending in the great questions. Thanks for playing along, those who join us in Slido for the quiz and our poll questions. Always good to keep things interactive. Josh, I want to give a huge shout out to you. Thanks so much for sharing the work you're doing and also why it is so important. And then I'm going to throw things back to Hannah before we wrap up for today.
Thank you.
Yeah, thank you so much, Josh and Joe. That was very interesting to learn about all the biodiversity Flower Garden Banks has to offer. So we are going to wrap up today's live interaction with a few housekeeping items. So this live interaction, as well as all of the other live interactions that Sanctuaries and Exploring by the Seat of Your Pants have partnered on, are on Exploring by the Seat of Your Pants YouTube channel in a playlist called NOAA's Office of National Marine Sanctuaries. So you can find all of our recordings, including this one. Give us a few days to get it uploaded, but all of them will remain there. I also want to promote our upcoming live interactions with Exploring by the Seat of Your Pants, including the one that's just this Friday, again in Flower Garden Banks National Marine Sanctuary. This will be another leg of the Flower Garden Banks expedition, and we will be discussing black corals. So if you found this interesting, please tune in on Friday. And next week on June 10th, we are kicking off World Ocean Week by showcasing our 360-degree videos of our national marine sanctuaries. So we're joining Joe at 11 a.m. with our videographer to talk about what it takes to capture a 360 video. We're going to show you a 360 video and then give you all the resources to watch them yourselves. So this will be a really exciting one to kick off World Ocean Week. More live interactions, if you have enjoyed this one, include all of our live interactions that I've mentioned thus far, in addition to our webinar series, which is geared for educators. We also promote Exploring by the Seat of Your Pants, as they are a great platform to bring explorers and scientists right to you through your screen. And then NOAA also offers once-a-month webinars with NOAA Ocean Today. So all of those links you can find here. And then to conclude, following today's webinar, you will receive a survey link. If you are an adult and capable of completing this survey, I highly encourage it. This survey helps us guide our programs moving forward from topics to formats. Your advice and guidance is taken very seriously. So I want to highlight that survey, and it only takes about 3 minutes. So it'd be very appreciated. And I also want to thank you all for joining in today and tuning in to our live interaction with NOAA's Office of National Marine Sanctuaries and Exploring by the Seat of Your Pants. I want to thank Josh so much for an incredible presentation on his work in Flower Garden Banks. And again, thank you all for tuning in. This concludes today's webinar.
Thanks, Joe. Thanks, Hannah.